Today we will discuss about the effect of RMC-7977 on native model of PDAC. To understand how this RMC-7977 will respond on the actual PDAC models or actual patients affected by PDAC. So in this case they took the model that more closely resembles to the actual patients affected with PDAC. And first they did the survival test where they find out that when they use this RMC-7977, the survival rates are even 3 times higher compared to the traditional survival rates. That is, with this gemcitabine, which is one of the commercially available enzyme for the treatment of cancers. Its survival rates comes to be around 30, near about 30 days, whereas survival survival days when this RMC-7977 used comes to a maximum of around 180 days, and gemcitabine results is more closely aligned with the vehicle, that is it does not have too much impact. And when they check the other parameters such as tumor volume, the tumor volumes also found to be grow slowly in the case where RMC-7977 is used compared to the vehicle case. In the vehicle case, the tumor volume growth occurs in a less number of days, whereas in the case where RMC-7977 is used, the tumor volume takes a longer time, indicating that the RMC-7977 cause tumor suppressions. And these things are presented in another form in the water plots, where we can say that, or in the bar plots, where we can see that when in the case of vehicle there is an increase in the tumor volume, whereas in the case of the RMC-7977 there is a decrease in the tumor volume from the baseline. After validating that the RMC-7977 has a positive impact on the native model of PDAC, they did the experiments on two different kind of mice at two different institute, and both of them are affected with different kind of uh, mutations. For experiment one is done at this KPC Y mice that has the CP C57B BI/6, and it is infected with this RMC-7977 at a dosage of 25 mg per kg every day. And these are the points where measurements are done, the blue lines and the green lines. And what they have find out is that in the first conditions, in the first experiment, that there is a tumor volume increase when in the case of vehicle, whereas there is a tumor volume decrease in the case of RMC-7977 treated till 8 days. After that there is a slight increase. And in the case of bar plots, the percentage change in the baseline is high in all the cases of vehicle treated mice models, whereas in the case of RMC-7977, when most of the cases the tumor volume goes down. And in terms of the side effect, they check the body weight, change in the body weight, and the body weight change is not statistically significant between vehicle and RMC-7977. And for the other experiment that is done at other institute, where they use KPC7 mice that is enriched with 129S4, and here they use 50 mg per kg dosage at alternate day. And in this case also, what they have find out is that in the case where RMC-7977 is used, there is a decrease in the tumor volume compared to the case of vehicle, and similar results are observed for this similar results are shown in the bar plots or water plots, where we can say that RMC-7977 treated cells has tumor regressions compared to the vehicle, and there is not much statistically significant changes observed in the body weight. So these two similar results further substantiate their point that RMC-7977 has a better response on the PDAC compared to the other available models. And in order to check the specificity of this RMC-7977, they check the other things such as apoptotic factors and phosphorylation or the PERK pathways. And uh, in this, and in the panel L, first figure of the panel L, they check about this PERK phosphorylation, and what they find out is that PERK phosphorylation goes down after 4 hours in all the 3 cases compared to the and PERK phosphorylation goes down in all the 3 cases, and it recovers after 24 hours. And this indicates these results are in line with the results that they have observed when they did the experiment on this KPC mice models that we have discussed in the previous video. So this is also a positive sign that the RMC-7977 gives the similar results that they observed in the mice models. And in terms of this phosphorylation of this S6 at S235 and S236, they observed the similar results. That is, at 4 hours there is a decrease in the phosphorylation of the S6, and after 24 hours they see the recovery in some of the cases. So in the most prominent in the 1WK case. When they check the apoptotic factor, that is CC3, in this case also they see that apoptotic factor goes up after 4 hours in the case of SD, and also in the other case, and it will go down after 24 hours, indicating that with subsequent dosage we can see the increase in the apoptosis and the tumors. And when they check the proliferations markers, that is cyclin A2, and they see that proliferation markers goes down after like 24 hours. Its effect is slightly delayed, that is after 24 hours. And these things are also in line with the previously observed results that they have seen in the mice models who are infected with RMC-7977. So all this data indicates that RMC-7977 has a better impact or better efficiency on this PDAC, that is pancreatic cancer cells, and with limited side effect or very minimum side effect that can be seen from the results of these apoptotic factors, and also from the results of these PERK phosphorylations. And the other data are shown in the supplemental figures, where they have shown that the effect of RMC-7977 has minimum on the colon and skin tissues, indicating that it has minimum negative impact on the healthy tissues. So I hope you understood what are the results that they have presented and how they come up to a point that RMC-7977 has better results on this PDAC, that's pancreatic adenocarcinoma diseases or pancreatic cancers. If you want to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.